Hi, I'm Carol Stepien. I'm the director of the beautiful Lake Erie Center, and our mission is to do research and address environmental problems in our backyard, which is the wonderful Lake Erie ecosystem. So we run a number of key research, education, and outreach programs in the Great Lakes. So we specialize in addressing environmental problems that are right in our backyard to improve our local ecosystems and ecosystem services. That are the things that humans get back from the ecosystem that affect our everyday lives.
My name is Dr. Christine Mayer. I'm a professor in the Department of Environmental Sciences at the University of Toledo. I'm an aquatic ecologist. People in my lab, students in my lab, are working to answer questions that are of practical assistance to supporting decision making for management and conservation in Lake Erie and the Maumee River. Several of the projects that we're working on recently are geared at quantifying how many fish of important species like walleye and perch are present in the lake. We've been doing work looking at connections of those fish and the habitat that they require, how that connects the lake, the river, and the shore. So, for example, one project we've recently been working on with a PhD student who just finished up here is looking at how fish growth responds to changes in water quality, turbidity. So events like algal blooms probably have the effect of reducing fish growth. You know, you can imagine if you're a fish and you're swimming around in a soup of green algae, that has an effect on how easy it is for fish to find food, and we see that playing out in terms of our model predicts what we see fish growth actually occurring in the lake. Another project that we've recently worked on in collaboration with partners at the Department of Natural Resources, Division of Wildlife, is looking at how people treat the shoreline and how that affects the fish community in the very near shore area. So we can see the effects of armoring the shoreline. So that means putting hard structures on the shore like riprap or metal walls. But the other thing that we can see there is that if people just let natural shoreline vegetation grow in that area, that we see more species of fish using that habitat. So that's a very positive thing, that information we can provide to management and conservation agencies about an action that people can take to help improve the fisheries community in their local area, right offshore of their own property.
My name is Dr. Tom Bridgman, and I'm an associate professor of ecology in the Department of Environmental Sciences and at the Lake Erie Center here at University of Toledo. In my laboratory, we're concerned about the environment of Lake Erie and environmental problems, and so we spend a lot of time out on the water taking measurements of water quality, particularly harmful algal blooms and water chemistry and oxygen levels that might influence the dead zone. And since we can't be out there all of the time, right now we are concentrating on putting instruments out there that will take measurements for us. So we are deploying instruments that will measure phosphorus in the water and dissolved oxygen near the bottom. And we'll put those out in May here, and we'll leave them out all summer long. And that's what we're focused on right now. And after we get those instruments in, we will be out on the water on our boat taking measurements probably every week throughout the summer.
Carol Stepien. I'm director of the Lake Erie Center for UT, and I'm also a distinguished university professor of ecology. I run my Great Lakes Genetics and Genomics Laboratory. I established this laboratory 2 decades ago to specialize in genetic issues in the Great Lakes with invasive species and native fisheries. And so what we do is we do genetic and genome tracking of changes in consistency in environmental patterns and the gene pools of fishes and invasive species. And here in the Great Lakes, we specialize on some of the biggest fisheries in Lake Erie, which are walleye, yellow perch, and smallmouth bass. We've worked on invasive species, including dry-seeded mussels, zebra and quagga mussels, the Eurasian rough invasion, the Eurasian goby invasions, the round and the tube-nosed gobies, and now we're working on Asian carps. So we've developed a new environmental DNA assay to rapidly detect all invasive and native species, identify their specific species identity, and also tell their genetic diversity and relative abundances. We've also worked on the VHS fish virus, which is like Ebola for fishes and first emerged in the Great Lakes about a decade ago. So we saw big fish die-offs here in Lake Erie in 2006 when we were out of the field. So we developed a rapid assay for VHS fish virus, and now we've been studying the evolutionary patterns and its genetic changes and how our fish populations have adapted to it, and we're trying to predict what may happen in the future. So a lot of our fish test positive to the virus, but now are not showing symptoms of it or showing relatively minor symptoms of it, and yet the virus is continuing to evolve or change with time. The newer things that we've been doing too is studying the genetic history of species in the Great Lakes. These are walleye scales. They were taken years ago, and so what we do is years later we use them for a different purpose. So the fishery agencies have already aged the fish, and so they save the fish in these envelopes, and so what they do is they give this to us, and what we can do then is grind up the scales and get the DNA off of the mucus from the scales, and we can tell the genetic profile of those walleye from years ago. And then what we do is we compare the genetics of the fishery today, the specimens we capture at given times and given places, with what was obtained there in the past. And so we can go back decades then, which provides really important information for fishery managers to know how their fishery and how their populations of these species have changed over time.
My lab is mainly focused on using the tools of landscape ecology, large-scale tools such as spatial statistics and GIS to look at issues of invasive species or the distribution of native species. Most of the work that I do deals with the spread of invasive species and how that impacts economic factors. For example, I worked on a project dealing with spread of emerald ash borer that showed that it would have been worth billions of dollars to fight that introduction as soon as it was arrived. Since then, I just finished a Fulbright scholarship program down in the Trinidad and Tobago, where I looked at the spread of a marine mussel, the Asian green mussel. It has impacts similar to the zebra mussel, where it clogs intake pipes, so the Caribbean is being impacted by it. Power plants, desalinization plants are impacted by this mussel. So the question is, can we slow or stop the spread of these organisms, and what is the value in spreading those? So we've also done work on spread of fish disease, VHS, as well as other Great Lakes species. We've done work on the distribution of darters, fishes throughout the Midwest. We've looked at how sediment impacts yellow perch in Lake Erie, and also how to model and track the spread of arsenic through constructed wetlands.
All right, my name is Jordan Rothkar. I work with Dr. Daryl Dwyer in the Environmental Remediation and Restoration Lab. All right, so our lab is focused on a few things. We have a couple projects going on. One is we're looking at cleaning up water that's going into Maumee Bay, and we're using a multi-pronged approach to do that. So we are working on farm fields to clean up agricultural water, remove some of the nutrients from agricultural drainage. We're looking at putting in systems to collect sediment that is coming through drainage ditches and streams. that have nutrients and bacteria associated with them. And we're also looking at putting in wetlands that remove nutrients and bacteria and things like that and create habitat at the same time. And so we're doing that out along Maumee Bay and some of the agricultural ditches out there. Another area that we've been working on is called phytoremediation. So we're using plants and bacteria to remove contaminants from soil and water. We have a couple of sites in Toledo. One is the former Jeep plant, and we've created a bioswale to clean up water that is in and going into the river. And then we're looking at a manufactured gas plant in downtown Toledo and removing cyanide from water there.